Thank you very much. So I would like to talk about Committee 5, the protection of the environment. Committee 5 is the youngest, the smallest committee that is out there, but we have a very interesting collection of academics, of regulators, and individuals that are involved in research institutes, and so we think we bring a very good perspective to the issue of environmental protection. We have 3 new members this year. Unfortunately, not all of our new members are able to be here at this time, but hopefully over the next year you'll get an opportunity to visit with them. Committee 5 has as its mission, it is concerned with the radiological protection of the environment, and its aim is to ensure that the development and application of approaches to environmental protection are compatible with those for radiological protection of man and with those for protection of the environment from other hazards. And this gives us a rather unique challenge, and also I would have to say that when Committee 5 was created, it was not without controversy regarding the need for it, whether it was good or even necessary to consider protection of the environment within consideration of radiation protection. What happened with Committee 5 was that it was decided that there would be, in essence, 2 parallel pathways considered for radiation protection. The standard pathway that you should be relatively familiar with, where we look at planned, existing, or emergency situations, we look at radionuclide concentrations, and then using reference male and female or reference persons, we make an assessment of doses, limits, and constraints, and make a decision regarding any sort of public health issues or modifications to the releases. And the thought was, as part of C5, that if we develop an architecture that parallels that which is in place for humans, it's recognizable, it's defensible, and it's consistent with what has been done before. In lieu of the reference male and female, or reference person, we have developed reference animals and plants, and I'll talk about those for a couple of minutes. Rather than dose limits and constraints, we have derived consideration reference levels, which are a bit of a mouthful, but in essence, they're bands of dose rates, and I will talk about those as well. The basis for Committee 5 came about in the publication of ICRP 91, published in 2003, which reviewed the ethics and principles and recommending system for environmental protection. Now, Jacques Lachard mentioned earlier that radiation protection considers science, values, and experience. And the same thing goes to play when looking at protection of the environment. In our instance, we are focusing on non-human biota. We're considering their adequate protection on the basis of understanding what effects are out there in the scientific literature. We've developed these reference animals and plants for which we place our architecture around, and then we let these reference animals and plants help us in assessing the exposure scenarios, developing concentration factors and dose conversion factors or dose conversion coefficients, helping us aggregate the effects data, and developing dose rate benchmarks. We also published ICRP 103. This came out in 2007. Again, looking at the aim is preventing and reducing the frequency of deleterious radiation effects to a level where they would have negligible impact on the maintenance of biological diversity, conservation of species, or the health and status of natural habitats, communities, and ecosystems. And again, the concept of reference animals and plants. So the difference between radiation protection, where you're using the reference male or the reference female or the reference person, we're focusing more on maintenance of populations rather than being concerned about the health and safety of a single organism, a deer or an earthworm or a bee. We're concerned about this maintenance of biological diversity, and this is one of the key distinctions in the work that we have done. So in ICRP 108, which was published in 2008, we came up with 12 reference animals and plants, and so the intent was that we have a number of organisms that were common in occurrence around the globe, that they spanned a terrestrial environment, an aquatic environment, including freshwater and marine, that they had organisms that lived on the surface, organisms that burrowed, and the thought was that this was not going to be encompassing every organism everywhere, but it was sufficiently representative that we could develop the architecture. And again, examples available of these species worldwide. And also the intent was not to have so many organisms that we would be unable to complete the documents in a timely fashion. So 12 RAPs with various life stages associated with each of them. In ICRP 108, we reviewed the biological characteristics of these organisms, including their occurrence, their taxonomy, their life cycle, their lifespan, their reproductive strategy, physiology, ecology, all of these things, again, with the intent to help build the structure for determining radiation doses to the organisms and at what levels that you might observe an impact. And again, in 108, what we did is we developed a methodology to calculate dose to the RAPs. And for those of you that are sufficiently gray, I should say, you may remember ICRP 2 when they looked at ways to calculate doses to spheres of tissue. Rather than looking at individual organs, what we did was we developed a series of ellipsoids, and the little cartoon shown in the bottom left-hand picture was introduced to ICRP back in the first meeting of our group as how ICRP Committee 5 views the world. Our organisms are these ellipsoid trees and ellipsoid worms and such, but it makes calculation of dose fairly straightforward. And again, these are fairly simple geometries, and they're analogous to what was done in ICRP 2. What was also important in the process was that we find information on radiation effects on these organisms and aggregate that. And what you see in front of you in this particular graphic are the bands called the Derived Consideration Reference Levels, the DCRLs. And we went out into the literature and where we could find this information, develop these exposure bands where it appears that there might be some probability of effect if you're seeing an exposure in the range of the band. Now for reference, there are some other comparison to other standards. You'll see for vertebrate benchmarks, generic benchmarks that have been developed by other organizations elsewhere in the world. In assembling this information so that we could develop these derived consideration reference levels, as I said, we went out into the scientific literature, and one of the things that we found as we were developing this architecture is that there really is several gaps in our knowledge and our information on radiation effects. And so in some instances, these bands represent the lower limit where we have any sort of information that's available. In being able to estimate doses to the organisms from radionuclides in the environment, we also needed concentration ratios. And ICRP 114, published in 2009, looked at developing concentration ratios for 39 elements and 12 RAPs with statistics based on the existing field and laboratory data, used new methodology to derive data, surrogates, if you will, where information was missing, and taking into account, again, the life cycle stages and habitats whenever possible. And then also discussing the robustness of the data. And again, in this particular process, we recognized, we came to realize that there really still are some substantial data gaps in our knowledge of radionuclide transfer from the abiotic to the biotic environment. In addition to the architecture that we've created for the RAPs and doing the dose calculations, we are now, in conjunction with Committee 4, publishing ICRP 124, which is looking at applications in planned exposure situations. And again, we are concerned with planned, existing, and accidental situations. And the question that you have to ask is, if you have some reference point where you have an existing concentration and you're interested in possibly optimizing it, you need to understand what the reference level is. Similarly, when you look at an existing exposure situation where you're trying to drop the concentration down, you have your DCRL that will help you make a decision as to what would be an appropriate level of exposure. And then finally, under an accidental situation, you have to consider what the potential impacts are when you have a release where you may have, for a brief period of time, a very elevated level of exposure, and then it drops back down. So again, the architecture that's been developed through the works of Committee 5 can help in that process. So just to sort of summarize where we have come from, we have publications 108, 114, and in press 124, and it enables us to take releases into the environment and make an assessment of what the internal tissue concentration might be in one of our representative organisms, using the dose coefficients, make an estimate of the dose rate to the organism, compare that to the DCRL, make a determination of potential impact, and then a decision can be made in terms of what the exposure might be. So we have this architecture that we've created, and the question now, well, where do we go from here? We've built this structure, we've published these reports. Should C5 be done? Call it a day. And what we have found in the process of assembling all of this is that we have seen many data gaps and process gaps that are in the scientific literature. And in the process of developing these reports and making this knowledge known, there have actually been a number of activities that have been initiated worldwide as a consequence of the work of ICRP. Now, we'll talk a little bit in our session on Thursday about some of the future work that we're looking at, going from the simplified ellipsoid models for calculation of dose, taking what we've learned from human dosimetry and building voxel phantoms and using that to do a better estimation of dose. We're also trying to move beyond the equilibrium calculations and do dynamic transfer modeling. So we're moving away from steady state. And this is really relevant in situations of emergency conditions or where you have pulsed releases where you're not at an equilibrium condition. Spatial and temporal factors in dose. What do you do for an organism that has a very large home range that moves across an environment that has a substantial gradient in the dose rate? How do you calculate dose? How do you determine an effect? That's a question that needs to be considered. And then simply filling RAP-specific data gaps. The effects data is fairly sparse, and we need to do a better job of trying to fill that. And then coming back and looking at those DCRLs in light of the data that we have, do they need to be adjusted? So in looking ahead, we're looking at consolidating our system and databases, extrapolating from our reference animals and plants to reference organisms that are used in site assessments. We're developing a report structure on that. We're looking at building monographs of the specific RAPs where we can consolidate and update the data that's available, including things such as stable element ratios, organ and tissue densities, many other factors that will help in the process of calculating doses. In the monographs, as I said, we have learned from the development of the human voxel phantoms, and we have found that we can now start developing phantoms for a variety of the RAPS organisms using CT and MRI scans and in conjunction with codes such as MCMP to develop more refined estimates of dose from internally deposited radionuclides. And then one of the other questions that we have to address is how to take the RAPs that are relatively stylized and limited number of organisms, and take the RAPs and the derived consideration reference levels and extrapolate those to representative organisms that might be more appropriate for a particular environment that you're considering doing an assessment on, so that, again, you can do a better job of protecting the environment. We also want to look at the effects data, and one of the really interesting developments is the area of species sensitivity distributions, where you take a look at what is the probability of causing an adverse effect in a particular species or groups of species in an environmental setting, and can you collate this data and, again, make a better assessment of the potential for effect in an area where you have an existing release, a planned release, or under emergency situations. One of the other things that we would like to look ahead to is take the DCRLs, which are bands of dose rate, which conceptually are fairly straightforward, but they're not so easy to test against or measure against. And can we devise instead concentrations of radionuclides in the environment where you can specify summation of radionuclides at a particular concentration or no radionuclide exceeding a particular concentration, such that you can be confident that no species, human included, are likely to be adversely affected. So this is sort of a practical extrapolation of the DCRL, and this is one of the next things that we are considering. So in conclusion, C5, although we are the smallest and the newest and perhaps the most controversial, I think we've done some good in our brief existence. We've helped develop a robust system that is compatible with the radiation protection for humans and the environmental protection systems for other hazards that are out there. We are considering the environment in its own right. It's appropriate and facilitates communication, and it also, I think, helps with stakeholders. We believe the system that we have developed is simple to apply using our reference animals and plants, but it is robust enough that it can adapt to complex exposure situations. For the future, for this next term, we're going to look at consolidating our data, broadening the scientific basis for what we've been doing, and improve the applicability of the work that we've done. Thank you very much.